Let's take another example. So let's look at our negatively charged side chains, glutamate. So the structure of glutamate is, so again, we start with the fully protonated form of the molecule. And C double bond O, OH. So again, pK1 refers to dissociation of the hydrogen or the proton from the carboxylic acid functional group attached to the alpha carbon. So again, if we start with the solution of glutamate with a pH equal to 1, all of the protons are still attached to the molecule. We add base, and if we reach a pH equal to 2.19, it means that half of these, so if we have a certain population of these molecules, half of the population will now be deprotonated at the carboxylic acid functional group. So we have this structure for half of the molecules, while the other half still have their protons in the carboxylic acid functional group. So the other half is still in this form, the other half are converted at pH 2.19. So if we continue to add more base, more of this form will be converted to this until we reach the first equivalence point. At the first equivalence point, all of these would have been converted into this form. We continue to add more base. This time, the next pH is 4.25. So if we reach pH 4.25, the proton from the side chain carboxylic acid functional group will be the one removed. So at pH 4.25, half of the molecules will have this form, while the other half has a deprotonated side chain. So we have this form of the molecule. So again, at pH 4.25, half of the molecules have this form, while the other half has this form. And if we continue to add more base, more of this will be converted into this form until we reach the second equivalence point. At the second equivalence point, all of the glutamate molecules are in this form. We continue to add more base. So what happens? We reach a pH of 9.67. At this pH, half of the molecules are still in this form, while the other half have their protons in the amino group removed. So we have this structure for half of the molecules. Then we continue to add more base until we reach the third equivalence point. At the third equivalence point, all of these molecules are converted into this form, and we have the fully deprotonated glutamate molecule.